Hello, Dr. Alonso. How are you?
Hi, Keith. How are you?
Good to see you again. Thank you for joining us today to talk a little bit about your grant and answer some questions about it. Um,
Perfect.
I think your grant's pretty exciting with the Delta 24 virus. But
Yeah.
before we dig into it, I'm going to go ahead and turn it over to Melissa from The Cure Starts Now.
Good morning. Nice to meet you.
Nice to meet you,
I'm Melissa
Melissa.
Fleming. I am the director of the chapter in the Austin Round Rock, also known as Team Brock. It's in honor of my son, Brock, who battled in 2016. And we have three other families, Caitlin, Alexis, and Parker's families that have all joined us. But I'm glad to be here this morning. Pleased to meet you. Did want to announce that we are awarding you a grant for $100,000 for your project Delta 24.
Wow. Oh my goodness. I
It
don't
seems like a surprise, so that is great. But
Yeah, I've applied several times. I was not lucky actually until today. So I was thinking maybe, Uh, I was, we are very excited about this project. We think it has a lot of potential and a lot of potential to arrive to the clinic. But of course, you never know, no? I've been working for a long time with oncolytic viruses. Kids know me, for a long time. And so, I don't know what to say.
no, and we want to thank you. It's been 8 years since my son battled, and I will tell you, I'm just as determined today as I was then to fund research. We need researchers like you that are focused on this, and I just want to thank you. At the end of the day, Brock wanted to be better, and it's researchers like you that's going to help make that happen. And so, thank you for joining our efforts. Thank you for being focused on DIPG, DMG, all of our kids. I did want to go ahead and introduce Jim Detry with the Cure Starts Now, because I know he also wanted to mention a few things.
Thanks, Melissa and Dr. Alonso. It's great to see you. I'm so excited for you and your team. So, on behalf of, the Cure Starts Now is 50 plus chapters around the globe. And on behalf of all of us, and also this is part of the DIPG and DMG collaborative, which is 24 other foundations that are part of our efforts. And as part of the development team, we're so excited to raise money for the special people like yourself and your team that are making a huge difference. And, on behalf of all of us, just wanted to say thank you.
Bueno, pues thank you also for, for believing in our project. I know there are many researchers out there, no, many also from the States. And for us has been a long journey. We started from very, back in the day with very few resources, and we little by little have been building this program, and we are really excited.
We're very happy to award you this grant. This is also a grant that isn't, and this oncolytic virus is not unfamiliar to us either. It's something that we have seen in some of the previous works that we funded with Cure Starts Now. And this one kind of stands out. Now, with this in particular, the Delta 24, and I believe it's what, RC, or RCR or RCG.
RGD.
RGD variation of it. You're trying to effectively tweak it to make it more effective and to get this immune response. Can you talk to us about what you see as different about this and why you're focusing on this type of virus?
Okay, the thing is, I work with adenovirus a little bit by my biography. I did my postdoc at MD Anderson, and I worked there extensively in this virus in adults. So I wanted, because I thought it's not toxic, but it has some effectivity. We thought it was a good tool to translate it to the DIPG, and we did extensive work to demonstrate it. We could open a clinical trial here in Navarra for 12 patients. And from these 12 patients, this is back already 2009, from these 12 patients, 2 are still alive 5 years later. This was just a phase 1 trial. So of course you cannot take out much about efficacy, but because we are a small center, we didn't choose any kind of patients, any kind of patient that went into the trial. So to have at least a little bit of increase in survival, because we have like close to 18 months, 17.8. I know this is very modest, but at least it's something that we haven't seen in other trials. So we thought we have to keep looking. And so we started to look specifically in what are these tumors, beside the tumor cells, what is surrounded, and how we can play with it to foster a better efficacy for the virus. So nearly by serendipity, we find this target, TIM3. This is one of these immune checkpoints that have been used, but has not been very much exploited. And we find it that in the IPG, it was expressed not only in the tumor, the IPG tumor cells, they express this target, but also in the surroundings, in microglia, in macrophages, and these tumors are full of them. So we have been doing some combinations, and one thing that we came across is because there are antibodies out there for TIM3 that the pharmaceutical has, but we have talked with a lot of pharmaceuticals, because after demonstrated that this could be a good target, we thought this was very easy to translate, to bring it to the clinic, just this antibody itself. The thing is that all these pharmaceuticals, they are not really interested in translating this. They've been trying these antibodies in leukemias, myelodysplasias. So because it was not working there, they are kind of abandoning TIM3, but we really think it's really a good target. And we have a paper that was published last year, I think, where we demonstrated this. So we were looking for things that we could put into the virus that they can be expressed, and from the same therapeutic tool modulate all this microenvironment. So what we propose in this grant is to use nanobodies, which are antibodies very small, that they are produced by llamas and camelids, and they are for whatever reason, they are super small and they are very easy to put into the virus. So we have produced one of it that has allowed us to do all this proof of concept that targets the TIM3 in mice. And now we are producing the human one. So it seems that this combination is very interesting. We already know that TIM3 by itself is very interesting. And we know that the virus is very interesting. So the idea is, okay, you put the virus and you kind of shake these tumors, making the immune system to come. And the TIM3, expressing an inhibitor of TIM3, it's just gonna allow us to produce a good environment for the virus to be more efficient, allowing when the lymphocytes come, they are gonna give them like sustain. Sometimes when the lymphocytes start to do their effect, they kind of get tired, of doing their efforts. So with this, we will sustain, but also it will target all this microenvironment I'm saying, microglia, putting them in a state that will favor the therapeutic effect. This is a little bit the idea. Help the virus to do its thing, and because we found that this target is very expressed in the IPGs.
Yeah.
So both things combined, we think, can be a very good. And in any case, just these nanobodies by itself, in the case that maybe we see it's not so good with the virus, that it seems is good, we need these nanobodies by itself can be used, maybe put it in nanoparticles or deliver directly. So really, you will have there two different biological therapies.